Okay, guys, it's time to talk about the Hardy-Weinberg genetic equilibrium or Hardy-Weinberg problems or principle. This is probably the most difficult topic that we'll talk about in the second semester, so make sure that you pay attention and let me know if you have questions during my quote office hours or via Schoology. So the Hardy-Weinberg genetic equilibrium is just another tool that we use to predict frequencies of genotypes, alleles, populations, those kinds of things. So we've already used Punnett squares to determine, to predict probability of offspring. Now we're going to use the Hardy-Weinberg genetic equilibrium. We're going to use this to try to predict future generations. So not just one particular offspring, but many generations of offspring. So we're going to talk about gene frequencies. So when we talk about gene frequencies, we're talking about how many of each gene are in a specific population. So when we're talking about genes, a lot of times we're also talking about alleles. So when I say gene frequency, what I'm talking about is an allele frequency, which means what percent of the population has a capital letter and what percent of the population has a lowercase letter. Now, of course, we should know that every individual has two alleles. So we're kind of splitting that up into not necessarily what their phenotypes are or their genotypes, but how many capital letters are there in a population and how many lowercase letters there are in a population. Now, this principle, the equilibrium, states that the frequencies, these allelic frequencies or gene frequencies, will stay the same if there are 5 conditions that are met. Now, I'm not going to go too far into these 5 conditions because we are going to talk about them at length when we talk about evolution. But if these 5 conditions are met, that means that a population will remain in equilibrium. Equilibrium, of course, means it's not changing. Okay? Now, none of these conditions will actually be met in nature. But we have to assume that they're met in order to predict future populations, which sounds a little bit confusing, but again, it's just a tool that we use to determine populations. So first, in order for a population to be in equilibrium, there can't be any mutation. So those are the random changes in DNA sequencing. They don't happen very often, but they can't happen in order for something to remain the same, obviously, because a mutation would be something changing. Nobody can enter or leave the population. That means that nobody can immigrate, come in, nobody can emigrate, go out. It also means that there can't be any births or deaths, which obviously will not happen within a real life population. Mating is random, which means every individual has an equal chance to mate with every other individual of the opposite sex. This does not happen. Just with humans, for example, mating, we have geographical mating. You're only going to mate with people who live in a particular area. Each animals also have, they have particular things that they look for in mates. For example, peacocks, the females look for a male peacock with like the brightest tail feathers. And cardinals, the females look for males that are the brightest red. Those are things that make mating not random. Population is large because small populations are going to be affected by chance occurrences, something like a flood, earthquake, something that can't be predicted. Those kinds of things are going to affect small populations a lot more than large populations. And then, of course, there's no natural selection. The environmental conditions have to remain the same, and every individual within those environmental conditions have to have an equal opportunity to survive. So, of course, we know that all of those can't happen, but again, we're using those Hardy-Weinberg conditions as a tool to look at what allelic frequencies are and how they change from generation to generation. So we're analyzing population to see if evolution is occurring. Microevolution, you should know the prefix micro means small. So all that means is gene frequencies change within a population. So the gene frequencies themselves are not remaining the same. And then to find out and then we can use them to find out percentages of genotypes in a population. So this is what we're going to be doing in this chapter. We're going to be doing this a lot more later on when we actually talk about evolution. But as far as this unit goes, we're really going to be doing this. We're going to find out percentages of genotypes in a population. So we use variables like you use in math class. The variables are always the same. P equals the percent of the dominant gene in a population. So I like to model this as, imagine that you have a population. So let's say it's our class, and we're all in the classroom together, and we're looking at one specific trait. And we're going to take all of the letters for everyone's genotype. So everyone has 2 alleles for each trait, right? So imagine that all of those alleles were just put in a hat. And then you are going to reach in your hand and pull out one of those letters. What would be the percentage that you would get a dominant letter versus the percentage that you would get a lowercase letter? That's what P and Q stand for. So if you're reaching into that hat, P is the percent of time that you're going to get the dominant or capital letter. Q is the percent of time that you're going to get the lowercase letter or recessive gene. Now, because there's no other options, you're either going to get the dominant or recessive. There's no in between. P and Q, written as decimals, if you add them together, is going to equal 1 or 100% of the population, which makes sense, right? Because you're either going to get a dominant letter or a recessive letter. So if you are getting the probability of the dominant gene and then the probability of the recessive gene, well, there is no other probability. So those two things together have to be 100% of the time. Now, we talk about P squared. These two we're referring to alleles, single letters. These are referring to actual genotypes. P squared would be the probability of getting a capital letter times a capital letter. Now, what is A times A? It's A squared. So that's why we represent, if P is the dominant gene, then P squared is P times itself, which gives you the probability of picking out 2 separate capital letters together. Q squared is the percent of the homozygous recessive population. So that's picking out a lowercase letter and then picking out another lowercase letter. So that would be, again, it would be lowercase times lowercase. Again, 2 things combined, it's squared. 2PQ is the one of heterozygous individuals. Now, P and Q make sense because you have a capital and lowercase letter, right? So that's your heterozygous individual. Now, you have to multiply that by 2 because there are 2 ways that you can get the heterozygous individual. You can get a capital and then a lowercase letter, or you can get a lowercase and then a capital letter, okay? So again, you have P squared, homozygous dominant in a population, Q squared, homozygous recessive, and 2PQ are the heterozygous individuals. Now again, homozygous dominant plus heterozygous plus homozygous recessive, well, there are no other options, right? Which means that is going to equal 1 or 100% of the population. I want you to set it up like this, okay? So it's a little pyramid. You have 2 boxes at the top split into P and Q, and that equals 1. And then you have 3 at the bottom, P squared, 2PQ, Q squared, those all equal 1. So theoretically, if you have almost any one of these variables, you can figure out all of your other variables. So anytime I give you a problem like this, I'm going to go back 1 second. So here's an example. Having a widow's peak is dominant. I'm going to set up my pyramid here. And again, P, Q, P squared, 2PQ, Q squared, just so that you don't get confused. Now, don't get thrown off by whatever the actual trait is. If you don't know what a widow's peak is, you're like, I don't know what that is. It doesn't matter, okay? I'm just telling you that it's dominant. which means having it is dominant and not having it is not dominant. If you want to know what it is, it's when your hair comes down to like a little point in the middle of your forehead, but it doesn't matter, okay? I am telling you that 16% of the population is homozygous dominant. Now, if I'm talking about population, homozygous dominant, genotype, phenotype, something like that, I am starting you down here, okay? And one thing I am never, ever, 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 ever gonna ask you to start with is this one, not 2PQ, because you can't separate out the P and the Q. So you've already have it narrowed down. I'm telling you that 16% of the population is homozygous dominant. So because I said population and I'm giving you a genotype, you know you're gonna be starting down here. And it's not 2PQ. So what you have to do is, well, let's figure out, is it P squared or is it Q squared? This is dominant. Therefore, 16% of the population, that is P squared, and we're going to convert the percent into a decimal, 0.16, okay? So that means that P squared, the probability of someone in the population being homozygous dominant, the probability of that is 0.16, or 16%. So based on this, 0.16, we can take the square root of 0.16 to get P. Now, keep in mind, we're doing decimals now. So P is actually going to be a larger value than P squared because 0.4 times 0.4 is that the numbers get smaller when you're doing decimals. So you're taking the square root of 16 is 4, therefore the square root of 0.16 would be 0.4. Now, if P plus Q equals 1, then 0.4, 1 minus 0.4, Q has to equal 0.6 because the two of them have to equal 1 together. Okay, now you can get Q squared because you have Q, 0.6. So now you take 0.6 times 0.6 and you get 0.36. And now you have two different ways you can get 2PQ. You can do 2 times 0.4 times 0.6. Yes, 0.4 times 0.6 is 0.24 times 2 is 0.48, or 1 minus 0.16 minus 0.36 should give you exactly the same thing. So again, another example here. Don't worry about what mid-digital hair is. Nobody cares, all right? All I'm telling you is the frequency of the non-mid-digital allele. So non-mid-digital, so if mid-digital is dominant, non-mid-digital has to be recessive. Okay? And then here's your key word here. I'm giving you an allele, which means when you come down here and do your boxes, if I give you the word allele or gene, I'm talking about P or Q. In this particular situation, I'm talking about the recessive trait, the recessive allele, so therefore Q is 0.4. Once you know Q, 1 minus 0.4, 0.6. Look, it's exactly the opposite of the one we just did. 0.6 squared to get P squared is 0.36. 0.4 times 0.4 is 0.16, which means again, 2 times 0.6 times 0.4 has to be 0.48. Yay! And there we go. All right, so we have the ability to roll the tongue into a U shape. That is dominant. Individuals who can't do it are considered recessive. So now we're not going into, I'm not giving you a percent, I'm giving you actual individuals, okay? 360 out of 1,000 people are non-tongue rollers, which is the recessive trait. So how are we going to get to the percentage of people who have that trait? Well, it's simple. We just take 360 divided by 1,000 times 100. And we get 36. That means 36%. Okay? Now, 36% of individuals in the population. Individuals, that means we're looking at either p squared or q squared. All right? Because we're looking at individuals. We're not looking at individual letters. We're looking at two letters together. So, we know that 36% of the individuals have the recessive trait. So, here's our little box. Which one is this? Well, they have the recessive trait. So that would mean they have to be q squared. So that is down here, and we're going to put 0.36 because that's the frequency of 36%. Now, I'll give you guys a little tip. Q squared is often the easiest variable to figure out because anybody who's q squared, they have to have lowercase letter. Somebody who has the dominant trait, they can be p squared, or that's 2pq, so they can be big, or they can be big, little. So q squared, if you're given individuals, is going to be the one that you're going to figure out if you have individuals or phenotypes or a population, something like that. So here we have 0.36. In order to get from here to here, we're going from q squared to q. We take the square root of 0.36 is 0.6. And then, hey, look, it's the same numbers as we've done before, okay? This has to be 0.4. This has to be 0.16. This has to be 0.48. Why do I keep giving you the same numbers over and over again? Because I'm coming at this in a different way, okay? I'm asking you the same question in several different ways. You have to be ready to answer them no matter how I ask the question, okay? So you have to know if I give you an allele, dominant, recessive, am I asking P or Q? You have to know if I'm giving you individuals, am I giving you P squared or Q squared? It's never going to be 2PQ, all right? Am I giving you the actual probability, or do you have to calculate the probability based on numbers that I give you? You have to be ready for it all, okay? So based on this information here, now the question is actually asking us for percentages. Well, the percentages are just your probabilities times 100. Okay, so the percent of the rolling gene is dominant, so that would be 0.4 times 100, which would be 40%. The percent of the non-rolling gene, again, gene, think allele. That's one letter. It's either one dominant letter or one recessive letter. No combinations. And then homozygous rollers, 16%, heterozygous, 48%, and homozygous non-rollers would be 36%. So guys, the hardest part about these Hardy-Weinberg questions is figuring out what variable I am starting you with. So I'm going to tell you that most of them are going to be word problems. If I say something like gene or allele, I'm talking about P or Q. They're single letters, either one capital or a lowercase, okay? But that's only if I'm talking about genes or alleles. Now, if I want something in the bottom row, I'm talking about genotypes, individuals, people, a phenotype, anything that is more than just one gene or one allele, we're going to be talking about P squared or Q squared. Like I said before, I will never, ever, ever begin you with 2PQ because there's no way to separate out the P and the Q. So what you have to do in reading these word problems is you have to be looking for those key words. Gene or allele is going to indicate that you're looking for P or Q, and then you just have to determine if it's dominant or recessive. Anything else, if I give you what an individual looks like, if I give you a phenotype, if I give you a genotype, if I give you the number of individuals, anything like that, well, if it's an individual, that individual has a gene, and that gene is two alleles, which means that it's going to have to be P squared or Q squared, and then you're going to have to figure out, well, is it dominant or recessive? I will give you another tip. Q squared, if I'm giving you individuals, 99 times out of 100, I'm giving you Q squared because the dominant individuals is P squared plus 2PQ, right? Because big and big little are both going to show the dominant trait, which means the only way that you're going to know for sure is by figuring out, well, who has that recessive trait? And if they have the recessive trait, then you know for sure they're little.